change, but it's about other stuff that I really care about, which is ecology. Yay! And we have two real scientists here in this session, and two more the next session. Our first real scientist is Brent Sinclair, who's going to be talking to us about plasticity, variability, thresholds, and evolution, and predictions.
Cool. Yeah, so thank you. It's a nice selection of people. If I stand over there, then I'm just going to drive you crazy because you're trying to make a film of it, so I'll stand here. So this is not the kind of place I normally hang out. I did 2 undergraduate philosophy courses and actually did surprisingly well, but I think that was more luck than design. And I'm going to say many things that philosophers will find completely egregious, so you're just going to have to live with that. So what I wanted to talk about, though, is about what happens with these models. So the conference itself is about climate modelling and the issues surrounding that. And when you've got a climate model, it's actually not, from a biologist's point of view, it's not very useful. What I want to know is how that climate, how the organisms out there in the real world are affected by that model climate. And what I'm going to do is basically go through and show you about all the things that my colleagues do that I think are wrong, and then why I think they're wrong, and then not come up with a satisfactory solution. Which I think is the same, is similar to the talk that we had just before lunch. We're talking about definitions of climate change. So I am feeling like I'm in a comfortable zone here that I'm going to do the same thing. So first thing, I work on insects, and insects are important. Okay, I have some examples here of some of my favorite insects. This is cabbage white butterflies. This is representing both pest insects and invasive species. Insects perform ecosystem services. This is a dung beetle. If it wasn't for dung beetles, then we'd be waist deep in Africa within about 6 months. We have insects of medical importance. This is a Rotman, a kissing bug that spreads Chagas disease. And there are also insects like these weta in New Zealand that are of conservation importance, so biodiversity importance and intrinsic value, rather than doing something necessarily for us. Now, insects are, or all of these insects are, and insects in general are almost all ectotherms, so their body temperature more or less reflects the temperature of the environment that they're in. They can do some little things, you know, one way or another. They can do some behavioural thermoregulation. There are species that will absorb solar radiation, and there are some species that can actually heat their bodies. But to all intents and purposes, we can basically take a temperature of an environment, and that's going to be roughly what the animal experiences. So people worry about insects because they're important and they're ectotherms. And this is just a random list of things that climate change is doing to insects. And you don't need to pay attention to those, but there's a lot of them. And one of the things that you should pay attention to is that many of them are not mutually exclusive, and some of them go in opposite directions. And so the challenge is to figure out, for your species of interest, let's say you're interested in Rotman here, what of these things is actually going to happen to the species, and what the impacts of climate change are going to be. So there's several different ways that people approach this with insects, several fairly standard ways. The first one are these correlative climate envelope models. So the famous one at the moment is Max Ent. I've worked with Robert Anderson, so I thought I'd better put his picture up there. And what we essentially do there is we take all of the distributions of the animal, of the species that we know of in its native range. You use that to say, on average, I'm already screwing up the video thing, aren't I?
Walk where you want.
Okay, I will. So you take these, you take the average climate or some measure of the climate in which you're finding it, so it's the average temperature or the rainfall or lots of things all together. You make a big correlative model and then project that onto some new environment, so where is a species going to be invasive or going to be a crop pest or something, or you can project it onto a new environment temporally, which is what you're predicting is going to happen with climate change. So in this case we have, I think, we have a species of western corn rootworm, which is a pest of maize crops. And we can see that we're predicting that it's actually going to, the red here is sort of marginal habitat, but it's basically going to be able to move further into North America. I'm sorry that those are a little vague, but that is North America after all that. I have some colleagues that think that this is not the best way to do it, and they actually do some work with fruit flies, and they suggest that the best thing to use is actually just the extreme tolerances of the animal. In this case, they're working with, this is Drosophila birchii. It is a Drosophila of conservation interest, would you believe? It's actually endangered because it's restricted to specific forest fragments. And what they show here is that the little black dots on these maps indicate the known localities of the species. If they just take average temperatures, then basically they predict that it can live all over Australia, which is patently not the case. If they take these extreme tolerances of these species, then they get something that looks much closer to the actual distribution, which is just on the northeast coast of Queensland here. And then if they just make a blanket 3.5 degree increase in temperature, which is a pretty bad way to predict climate change, but we'll run with that, then they show that there's a decrease in the potential range of this species. In actual fact, I should add that actually this species is endangered because of forest loss and fragmentation, not because of the temperature at all. A different approach sort of premiered about almost exactly 5 years ago this week by Curtis Deutsch and colleagues. And what they did was they took the thermal performance curves of insects. So here we have temperature on this axis, and we have the relative fitness of the insect, however you want to measure that. So probably reproductive output for these numbers. And there's a lower bound, the critical thermal minimum, or a maximum and an upper bound here, and an optimum performance, and this sort of characteristic shaped curve with a convex surface on the side and a concave surface on that side, which has sort of mathematical implications, which I can go into at great length if someone ever wants me to. So what they then do is they then basically say, well, what is the distribution of temperatures, and are we seeing temperatures move in different places along this curve? And what they show in this paper is that basically if this is latitude here and change in fitness in 2100, what they show is that if you're in the tropics, you're already very close to this T-opt, which means that if the temperature increases a little bit, then suddenly you're scurrying down this downward curve, which is, the curve is asymmetric, so you're getting very close to your CT max very, very fast. So they predict that tropical species are much more vulnerable to climate change than our temperate species. Now, when I actually presented this in our lab journal club 5 years ago this week, and one of the things that I pointed out was that it was perfect for Halloween, because one of the scary assumptions they make is that there is no winter. They only look at the warmest 3 months of the year. They assume that the rest of the year doesn't really matter at all, and I'll talk about that. I should also add that Johannes Overgaard, one of the authors of this paper, has evidence, empirical evidence, looking at Drosophila species, showing exactly the opposite, that the tropical species are less susceptible to climate change. So, good for them, I guess. So basically we've got this problem where things can go either way, right? It depends. It depends on who you ask, it depends on how you interpret it. We've got these different models, and there's a whole bunch of assumptions going into them. So the first assumption is that the climate model is accurate, right? And that's what the rest of this conference is about. I'm happy to trust that. These seem like very, very smart people. They've got big computers and lots of nice glossy things, and I really like little circulation models and things. And that it reflects insects' experience. That's a little trickier because they're modelling climate on 100 kilometre grid squares, right? And there are no insects that are more than 100 kilometres long, I can assure you of that. And as a result, we're assuming that what they see at this 100 kilometre grid scale can then be narrowed down to what this little insect is experiencing. And that's a whole different problem. But you can argue that this max-end approach, this correlative approach, is actually a reasonably valid approach under those circumstances, because you say, well, the insect is integrating some of these average things, and that works okay. More worrying for me is that they're assuming that these insect input data are meaningful. And that's one of the things I'm going to talk about today. And the other thing is that it's focused almost entirely on the growing season. And I'm going to talk about these two things because I think that this is really important to consider when we're trying to make projections of what insects will do under climate change. And we care about this because we will get malaria, we will get West Nile virus, we will get Chagas disease, and certainly in the southern US. So it matters to know what these distributions are. Okay, so two important points on this slide. The first is that winter matters. Okay, so this is a life cycle of a eastern tiger swallowtail, maybe, some kind of butterfly. And in green here we have the growing season, so we have the butterflies fly around, they lay eggs, they're a larvae, they start off, they're really cute, they're like little bird poops, they grow up into these little things that have eye spots, and when you surprise them they stick out a tongue and look like a snake, And then they pupate, and this is the really interesting time of a really interesting life stage, because they spend 2/3 of their life as a pupa overwintering. Okay, so the growing season that Deutsch and co are interested in is this tiny little portion of the year, right, compared to the amount of time that they spend overwintering, which they completely ignored. Now, winter is not straightforward. It's easy to just say, oh, look, they're not doing anything. Look, it looks the same. It doesn't move. Therefore, we can ignore it or just assume that the changes will be negligible. And I don't think that's the case. So I've simplified winter conditions on, or the effects of winter conditions on insect performance here for you. So we have drivers here. I only put 3 drivers into this conceptual diagram to keep things straightforward. We have average temperature, we have thermal variability, and we have snow and ice cover. And we have processes that are affected by this, and then we have a whole bunch of biological impact. Now, the important thing to take away from this is that each of these circles indicates something that could go either way. So we spend an awful lot of time saying it depends, right? so winter matters, we know that it's really important, and now I'm telling you that we don't really know how it matters. Okay, so what I'm going to do for the rest of this is talk about what this means for our understanding of insects, because I'm really only interested in winter anyway. So, the first important thing to realize is that insects are diverse and they're not static, right? So these are just 3 of the species that we work on in my lab at the moment. We have a weevil that buries itself about 25 or 30 centimeters underground. It buffers itself from most environmental conditions. We have fruit flies that hang out in the leaf litter, so they're partly buffered, but they're also victim to all sorts of other things. And this amorphous blob here is one of the most awesome animals in Canada. It's the goldenrod gorefly, which is going to grow up into a very short-lived, cute little fly. At the moment, they overwinter as a pre-pupa. They are, I assure you, delicious because they have an unusual kind of fat and they accumulate a lot of glycerol and sorbitol. And they're exposed to the ambient conditions to the extent that these guys will have 100% survival at minus 40 degrees C, so they're frozen solid at that point, you can smash them. And they have measurable survival at minus 80, which, by the way, is colder than any temperature recorded in North America ever. Okay, so we've got all these complicated things, and the title of the talk is Plasticity, Variability, Thresholds, and Evolution. So what I'm going to talk about is the fact that the way that these different organisms respond to different kinds of conditions. But as I do this, you've got to bear in mind I'm going to show you lots of examples from lots of different species, and I think there's maybe 7 species in this talk, which means that we only need to understand these processes for about another 19,990,000 insects. So, if you thought the human genome was a tough project, try thinking about it when you're trying to extrapolate to all of these different genomes and all of these different environments. So the first thing I want to talk about is phenotypic plasticity, so variation within a genotype among species. And this is just coming back to Drosophila. None of these species of Drosophila are at all useful, I should add, but what they are useful for is that you can catch them easily and they don't fly that far, so these are distinct populations. So we have latitude here. These are studies from both Australia and India. And what we see is each of these little spots is a population of a species, and we can see that there's a general relationship that this is just one measure of how they survive, how cold tolerant they are, that as you get further from the equator, they become more cold tolerant. That makes quite a lot of sense. However, there's quite a lot of variation here. So, if I chose these 3 points, I could tell you that there is no difference in the cold tolerance of these 3 species. If, on the other hand, I chose these 3 points, I would be able to tell you that these species are completely different. Now, which one do you put in the model? Right? That's kind of difficult. And it gets worse, because these are different populations. There's probably some genetic differentiation between these populations. This is the same population of flies. Okay, so we collected them from my compost heap in 2007. We grow them in the lab. When they're outbred, they've got massive genetic diversity. When we want a bunch of flies, we just pull them out and do stuff to them. And what Victoria Ransbury did was she took these flies and she gave them lots of different conditions. She reared them at different temperatures. She gave acclimation treatments. She gave them cold shock treatments. She spoke to them sternly. You name it. And we've got 2 different measures of cold tolerance here. But what I want you to notice is that basically by doing these things, we can change the time it takes them to recover from the cold by 40 minutes. The scale only goes up to 50 minutes. Or we can change the temperature at which they fall over by 14 degrees C. That's larger than the entire range that we get across those species that we see in this diagram here. And in fact, it's equivalent to the range that we get, one of my colleagues in Denmark did a study with 99 species, and he gets about the same range among 99 different species of Drosophila as we can generate with one population. Right, so what do you put in your model? Okay, so other complicated things. The temperature does not remain constant, right? It varies. And one thing, if you ever spend any time in London, Ontario, is you know that we get this midwinter thaw. And so Steffi Sobeck, she's wearing a respirator here, she was allergic to the Emerald-Ash-borer, but she was working on these guys, and what she showed was that if you take these Emerald-Ash-borer in the winter, so this is you can't see that, but this is just the temperature they freeze and die at. And it's pretty cold, they survive well below -30. And you can stick them in the warm and they become less cold tolerant. If you then put them back under cold conditions, they can't recover that cold tolerance. So there's plasticity in that they will lose their cold tolerance, but they can't recover it again, so it's irreversible. There are other species for which it is reversible, so that's not a general rule. A different approach to variability, this is Caroline Williams, who did a PhD in my lab, and she was working with two different populations of this rather ugly looking caterpillar here. It grows into a really unexciting butterfly as well. And she was the only difference between these populations was the variability of temperatures, particularly during the fall. And what we see is she's predicting the amount of energy that they use over a winter across a large number of years, because we make a whole bunch of egregious assumptions. And basically what she shows is that in the variable environment, these guys use about 50% more energy than in a stable environment. Now what's different about these things is that these guys have the ability to suppress their metabolism, which makes most of that difference go away. These guys do not. So we've got a difference, an among population difference in the ability to do the stuff that I just showed that the flies do. This is Katie Marshall. I put this picture up because she knitted her own bouquet for her wedding, which I think is a skill that should not go unnoticed. But she was working for her PhD on this thing here, the spruce bugworm. It overwinters as a second instar larva. That's actually a third instar larva, so they're about 1.5 millimeters long. We can buy them by the thousand because they're a huge crop pest, so people rear them so we can order them in. And they do this sort of thing. So they will eat up to 50,000 square kilometers of forest in a year when there's an outbreak, and they have these cyclical outbreaks. Now, Katie's PhD thesis was looking at what happens when they're exposed to cold more than once. Because we do all these experiments, we take an animal, we freeze it, thaw it out again, see what happens. It turns out that the number of times that they're cold exposed is more important than the temperature that they're exposed to or the amount of time that they're exposed to cold, in terms of determining all sorts of things to do with their fitness, their reproductive output and their growth rate and things like that. So what she then did was, and this is work she's done since, is that she then took a bioclimatic envelope model, and as well as all the other average things, she also included the number of times that a temperature threshold was crossed, and she used that to make a model. Now, if you include that in the model, so this is her model of just a blanket 2 degrees C increase on climate norms. But basically, this is what the predicted distribution of outbreaks in a 2 degree temperature increase looks like without number of exposures in the model. If we include the number of exposures in the model, then this is what it looks like. So basically, depending on whether or not you think this is important, and we think it is because we have shown empirically that repeated cold exposure is bad, spruce bugworm is either going to be a problem or a multi-billion-dollar problem. And it's the kind of information that is useful to have and that we don't know. And this is one species that we know this for. Okay. Finally, the last thing that I wanted to talk about is the fact that evolution is happening. Because insects have a short generation time, evolution is happening at a time span relevant for climate change. This is work by Caroline Williams again on fall webworms. So these are these sort of things that you see around walnuts and cherry trees and maples and things in the fall. And these were wonderful to work with because each of those nests is the entire reproductive output of a female who had mated once. So we could do all sorts of cool things with genetics there. But what I want you to pay attention to, we have two different measures of fitness. We have survival over winter and development time. And we have two different kinds of winter, and we have two different places. So we have a warm environment and a cool environment. And what we see is that in some measures of performance, there's just a general thing where these guys are better than those guys. In other places, these guys are better than those guys, but only when they're under warm conditions. These guys can disperse, so it suggests that there's enough genetic variation out there that maybe we just get changes in the population associated with climate change, not a problem. The best example of this is from Bill Bradshaw's work, where they showed that the timing, the photoperiod response of pitcher plant mosquitoes to go into diapause has shifted over a 24-year period. So basically, they were responding to having longer growing seasons by remaining active for longer during the year, which is pretty interesting as well. Right, so it's complicated. Do we just give up? Well, I don't think we can, because this is urgent. It's nice to sit around and talk about climate models and things, but we can't wait until our knowledge is complete. We can't wait, and you can't just keep giving me money until we've done this for all 19 million insects or whatever, and I can give you a definitive answer. So what do we do? So I think that what we really need in terms of the insect physiology is some kind of predictive approach that allows us to at least categorize insects and say these groups of traits or these groups of insects are going to perform in these kinds of ways. So average approaches could be okay in some circumstances. It's what we've got, it's what can be done quickly. I'm interested in using approaches to identify species that are going to be susceptible to climate change and species that are going to be robust to climate change. And we're starting to identify some basic traits that we can do that. The other thing I'm interested in is understanding the mechanisms that underlie these responses, because I would like to be able to go out, catch a whole bunch of insects, grind them up, look at gene X, and then tell you those 3 species are the ones that are going to be susceptible to climate change. You need to do more work on them. Ignore those 500 other species. We can't do that at the moment. There is no a priori way to say this is a species that is thermally plastic and one that is not, for example. One thing that I'm starting to do, my wife is a conservation biologist, so she sort of is forcing me to also think about using physiology to inform conservation decisions. And I say guesses in that case because she will freely admit that that's more or less what they're doing at the moment. But from the perspective of habitat restoration, she needs to know what is going to want to live where in 100 years when the forest she plants is a forest. And that changes the decisions and it changes the trajectories that you're aiming for in terms of the species that you're planting. And I think that physiology and the mechanisms is a good way to approach some of that. So that, oh, this has stopped working. There we go. So that's all I have to say. Thank you. This is my lab and some of their assorted hangers-on. But the point is that they do all the work, I just kind of watch. And hopefully I've left a little bit of time for questions.
We have 7 minutes for questions. So
I have 2 questions, but one's really, really tiny. With the you had one graph near the beginning of your talk, a student that was looking at within a population, different tolerances.
Yes,
yeah, that one. Yeah. Is that within individuals, or is that like over generations?
No, so this is a single generation.
Okay.
That was split up.
Yeah.
But each of these points would be, I think on this axis it would be the average of 100 individuals, and on this axis it would be the average of probably 20 or 40.
So it's between individual within population?
Yeah.
Okay.
Hang on. Yeah,
okay.
I guess so.
So
Yeah.
the different individuals are doing different things?
Yes, but they all have effectively the same genetic, they're not inbred, but they have the same panmictic genetic background.
So then my question is, what do you think drives that difference? If they're all like sort of the same genetically, is it like an environmental effect?
It totally is. So this is just responses to different things. So we know, and this is where understanding the mechanisms can be kind of important, because there are some species that can't do this. I'm showing you the extreme case, right? There are some species, and there's a theory that I keep harping on about my friend Johannes for some reason today, but he hates this theory, but I think it's a really interesting one, which is that species that have relatively high thermal tolerances do not have very much plasticity left over. So basically they've spent all of their resources getting as, say, high temperature tolerant as they can be, and there's not very much space for expansion. And that's borne out particularly for high temperatures because there's actually very little variation among species in high temperature tolerance. So a species that has a higher basal tolerance just tend to have much smaller plastic responses. At low temperatures, it's a lot more tricky. We know a little bit about what these mechanisms are, but not as much as we probably should. right? So we can grind them up and we can measure stuff. My current favourite hypothesis is that the more cold tolerant you are, the more the less sodium you have in your bloodstream. I don't know, but it's the kind of thing we could measure easily. We could grind up thousands of animals and measure that across a huge range of distributions, and potentially, if that works, then that would be a great magic bullet for giving us an initial screening tool. Yeah.
So it just actually, this next question actually has to do with that. Another sort of screening tool, has anybody done sort of a phylogenetic analysis on like tolerance and plasticity? And like, are there any patterns there that you can kind of use as like a general guideline? Sorry, if anybody doesn't know, phylogeny is how different species are related to each other on like an evolutionary time scale.
Yeah, so that's a problem. Yes, it's an obvious thing to do, and yes, we've worried about it. But the 99 species study did take phylogeny into account, and we do that as well. The problem is that the timescale, even of evolution within the genus Drosophila, is much larger than the timescale of small climate fluctuations. So most insects have redistributed themselves poleward as the glaciers retreat. So essentially we've had sort of homogenizing selection a lot of the time, or we've had repeated radiation. So it's very hard to predict, and actually some work that I didn't talk about, we've been working on stick insects in New Zealand. So there's 23 species of stick insects. They all are descended from a single common ancestor from New Caledonia. And they've invaded the mountains on at least 4 separate occasions. They used 2 different cold tolerance strategies, and those are all underlain by 4 different genetic responses. So within 23 species, I can show you 4 different ways to skin that cat.
I have kind of a naive question, but it seems that you suggest plasticity is a problem when it comes to time to modeling response to climate change, in a sense. And I'm not sure I understand exactly why, so perhaps you can say a bit more about that. And my other question is, when you get to the end of your talk, you talk about conservation and restorations and things like that. Wouldn't plasticity be a good thing when it comes to time? Okay.
Absolutely. So plasticity is great. The problem is that people are not taking plasticity into account for the most part. So, for example, when you take your input data for your biochromatic envelope, for example, you don't know that those climate variables are actually what's constraining the insect, for example. The insect may actually be being constrained because of host plants, or because of habitat, or because of something else, or competition. The other thing is that, for example, the Deutsch approach with the thermal performance curves, which is, has been revolutionary for the field in terms of a way to explore this. But I was talking to Francesco about this earlier. The problem is it assumes that the insects don't change at all, right? So it's not so much that plasticity is a problem, it's just that you need to incorporate it. And because the level of plasticity is heterogeneous, you can't just assume that all animals would do the same thing. And the classic example, actually more for snails than for insects, but I do a little bit of work on snails. And snails just shut their metabolism down. They shut off they close their shell. Intertidal snails will actually perch themselves on the edge before they do that, so that they have the least possible contact with everything. And then they'll wait it out for 4 months if the weather's inclement. So high temperatures in the tropics are not a problem for them at all. So there's lots of things like that where there's actual state changes that can make a big difference, as well as just shifts in the as well as plasticity. So it's all stuff I think that is actually should be relatively easy to incorporate into a model. It just has not been explored very much. Yeah. So I didn't mean to imply that it was impossible to do. All of those things you can include. And in fact, just an example of that is that we actually included this deacclimation effect in a model of emerald ash borer distribution. And it made no difference because we never actually saw the combination of conditions that would be necessary to lead to mortality. So it's great that they do it, but it doesn't actually, in this case, it doesn't actually matter. Yeah.
Any other questions? Good. So I can ask a question. So I want to push back a little bit on the plasticity being sort of somehow not insanely hard to take into account.
Yeah.
Because, I mean, it is,
Yeah.
But most of the time, it just, I mean, how does it let you make any kind of predictions, which are anything but sort of like, whoa.
Yeah,
maybe
so
this is
I think the way to approach this is more sort of the approach that Katie took, which is, so she said like this is important. But I think it's actually to take a sensitivity analysis approach to these models, right? So you say, okay, if we have these kinds of plasticity, what does that mean for the outcomes of our models? And which things are important and which aren't? This is not important, other things might be. And so that allows you to start to define the traits that you're looking for that will determine susceptibility or tolerance.
So is there a limit on the sort of taxonomical scale of how far you can go? So I mean, I can see that it might work for a species or a genus.
Yeah.
But more than that?
Oh, absolutely, and there's always going to be exceptions. Like within Drosophila, we now have, so one of the things that I showed you is chill coma recovery time. You put the animals on ice and then you see how long it takes before they can stand up again. And we now have a really, really good understanding of what happens during that process and what causes variation in that process. Unfortunately, as far as I can tell, it's completely divorced from anything that's useful in the real world for understanding cold tolerance, but we'll leave that aside. So that's great, and we can do it, and we can do it for crickets, and we can do it for Drosophila, so we're getting pretty good there. Except that it's all about balancing sodium and potassium and the hemolymph and stuff, and caterpillars don't do it that way. Caterpillars have a completely different system of maintaining iron and water balance. But what's crazy is that butterflies don't. So even if we and beetles mostly do, except for when they don't. And when they don't, it's either because they live in certain habitats or because they eat certain things, but not both. So we can start to sort of say, well, okay, this would apply to everything except larval Lepidoptera, for example, which is actually making some progress. And we can say it applies to all Drosophila except Drosophila persimilis, which I wish I'd never let into the lab. Then we're making some progress, right?
Yeah. Let's thank our speaker.